All right. Are there any questions? Say everybody got homeworks number 2 and 3, right? I mean, and I put in the due dates. And, homework number 3 is very, very short, so it shouldn't even take you that long, right? And then after this, so homework number 1, 2, 3, then there'll be 2 more homeworks. Okay. Again, nothing really difficult because I've even assigned it to undergrad students before. Okay. So, and the exam is on November 3rd. Yeah, right? November? No, no. Yeah, it's on the Thursday, right? I mean, yeah, so just, keep on studying for that exam, okay? That's based on molecular biology. And this material on which you have turned in the homework, there's no exam on that, okay? There'll be one more exam on the plant biology stuff. But I'm still working on preparing notes and all that for that portion, okay? So, any questions on the material that we covered last time? So then let's continue with our discussion. And today I'm going to cover that other paper, right? I gave one paper, I think we call it paper number 2, right? And along with the explanatory material, I don't know whether I can finish all of it today or not, but I'll try to cover as much of that as possible. And you will be pleasantly surprised to see that, in that paper right now you have enough background to almost understand everything, right? After today's lecture. Today and maybe a little bit into the next class. All right, so this is where we had stopped last time, right? And the key message here was that nonlinear dynamical systems, they seem to provide the appropriate framework for studying genetic regulatory networks. Another characteristic that you'll have in genetic regulatory networks is that the control is distributed, right? So distributed control is common for complex systems which have the property that no single agent is singularly in control of the system behavior. Rather, control is dispersed among all agents with varying levels of influence, right? And this is the current view of genetic regulatory networks. And the take-home message here, right, is, before we get to the take-home message, right, again, if you have a network, right, that is modeling the interaction between the genes, right, you might want to push the network from some states that are associated with bad behavior to states that are associated with good behavior, right? So that is the connection. That is the connection between engineering and biology, right? Of course, there's a big gap here. Okay, how do you come up with a good network model, right? So we have some ideas about that, but we are nowhere close to the finish line, right? So that's why, I mean, there are several of you that are PhD students, even maybe advanced master students. So there are lots and lots of topics that you can work on, right? And this area is unique because, nobody can question why you're working on that, right? Like, the work that you will do, it is not just for getting your degree, right? But it will hopefully, get us closer to solving a societal problem, whether it is in agriculture, it's in cancer genomics and so on, right? And I mean, if you can do some good stuff, you can probably make a name for yourself and become extremely successful, right? So this is quite different from, let's say, working, let's say, in the controls area, right? Or in the communication area, where probably some of the hard problems are left behind. And even if you solve those, nobody really cares that much, because it may not make that much of an impact, right? It might be more of an academic exercise, right? So it is worth spending the time to learn all this biology, right? Because it opens up a whole array of problems, right? Does not mean that you're going to be any mathematically any less rigorous or anything like that, right? But it is going to be motivated by the real world application, so that if you succeed, right, with your math in solving that problem, right, the potential impact is huge. So to significantly change the global behavior of a system, right, in a desired manner via external control, it is necessary to consider the effects holistically, because you should not look at one gene at a time, right? Because the genes interact with each other in a multivariate fashion. So this is really a lot more complicated, and that's why a lot of times, especially in the case of cancer, the biologists might think that, okay, they found out this gene, they conduct experiments, right, and then they find out that it doesn't work, right? Because that gene in isolation is not what is determining the final fate of the cell or the tissue and so on, right? Now, again, I mean, for biological systems, they have some degree of robustness, all right? Because, again, I mean, there's a global stability of genetic networks in the presence of small changes to the system, right? Say, for example, your temperature, it has to be controlled within tight limits, right? Your blood pressure, right? So there is stability. I mean, just because the temperature outside goes up, right? Your temperature, body temperature, even if there's no air conditioning, right? As long as it doesn't go up too much, your body temperature will adjust, right? So the mechanisms are in place, right, for adaptation. So the previous issue about controlling a gene regulatory network, right? Here the issue of controllability will come up, okay, because you want to figure out, which gene in the network you can use, right, to achieve the desired objective, right? Again, I mean, this is not controllability in the sense of control theory, right? Because how many of you are familiar with the notion of controllability in control theory? Most of you are, if you've taken like 605 and other courses, right? You want to move a system from a state A to a state B in finite time, right? The ability to do that. So here also you need some kind of controllability, because if the gene, in other words, if the phenotype that you are trying to affect, right, is tone deaf to the perturbations in this gene, right, then you can work as hard as you want, nothing is going to happen, right? So that, again, that's also an open problem, right? I don't think that people have figured these things out, right? So in summary, right, our conclusion is that nonlinear dynamical systems provide a framework for modeling and studying gene regulatory networks, right? But I'm not making the claim that we have figured out, how to take each biological phenomena and map it into the appropriate regulatory network. If we could do that, right, then the tools are in place, right, in the engineering literature, right, because we analyze all kinds of systems, right, like traffic networks, right, then internet traffic and things like that, right? The tools are already in place. And for those of you that are interested, I would recommend that you take Professor Kumar's course next semester, right? He's going to talk about Markov chains and control of Markov chains and all that. So that is going to give you the stochastic background that you would need for uh, doing this kind of work. Now, so you're trying to model gene regulatory networks. So the key question that comes up is, which model should you use, right? Should you use differential equations, right? Should you use Bayesian networks, right? Should you use Boolean networks and so on? Okay, there are many models that are available, right? So model selection will depend on the kind and amount of data available and the goals of the modeling and analysis. See, here the thing is a little bit different. Even in an engineering system, there are probably physical laws that you understand, right? Newton's law, and even there you have to be careful. You cannot use a very complicated model to do a control system design, right? Because then, first of all, you'll probably only stare at the model, not be able to make any progress. Second thing, anything that you come up with, when you implement it in the real world, it'll probably not work, because there'll be a mismatch between the model and the actual system, right? So the goal is to try to use as simple a controller as possible, right? And I believe even in other engineering systems, it is a similar situation, right? So here the classical engineering trade-offs are: should a model be fine with many parameters to capture detailed low-level phenomena, such as protein concentrations and kinetics of reactions, but thereby requiring a great deal of data for inference? Because all these different constants that are going to appear, right, nobody's going to come and give them to you, right? This is not a textbook problem, right? So if you introduce more parameters, you have to figure out a way of estimating them. On the other hand, should the model be coarse, with fewer parameters and lower complexity, thus being limited to capturing high-level phenomena, such as whether a particular gene is being expressed, right, or not expressed, right, at a given time, but thereby having the advantage of requiring much smaller amounts of data, right? To learn this kind of model, all right, this is a coarse model. It's not going to give you all very detailed information, but on the other hand, to learn this model will require a lot less data, a lot fewer parameters to work with. Now, model selection needs to obey what is called the principle of Occam's razor, right? It basically says that model complexity should be sufficient to faithfully explain the data, but not be any greater, right? Don't make any more complicated a model, right, than you need to explain the data, right? If your data fits a straight line, right, don't put in like cubic splines and all kinds of things, just to appear mathematically sophisticated, right? You think if you're presenting something, you're going to impress it, but in the real world, it doesn't work that way, right? So it should be as simple as possible. So from a pragmatic engineering perspective, this is interpreted to mean that the model should be as simple as possible to sufficiently solve the problem at hand, right? Now, in the context of a functional network, right, how are you going to, attribute complexity, right? Complexity would probably be determined by the number of nodes, number of genes that you're taking in this network, right? The connectivity between the nodes, right? How the nodes interact with each other, right? And the complexity of the functional relations, and also the quantization. Are you going to do binary quantization on and off, right? Or are you going to quantize it to 5 different levels, right? Or are you going to, if you work with analog, you'll need a lot more data, okay? So here we are going to focus on the original deterministic version of what is called the Boolean model, right? And in the Boolean model, see, if you have n genes, right, in a Boolean model, right, you have n genes. The update of each gene, right, the activity status of each gene at the next time point, right, is going to be given by a Boolean function of all the variables that affect it. Right? That is the key defining property of a Boolean network. And the Boolean networks were introduced in the 1960s by a guy by the name of Stuart Kauffman, right? And since then they have been extended to the stochastic case, right? Because you might say that the transitions are deterministic. Like at node number 1, I know that the update takes place according to some particular Boolean function involving nodes number 3 and 6, right? But you may not be 100% sure, right? So you might say that, okay, at node number 1, the update takes place according to the function number 1 with a certain probability, function number 2 with certain probability, function number 3 with certain probability, right? So that way you're allowing uncertainty. And if you do that, I'm going to show you later that then what happens is that stochastic Boolean network, right, it can be modeled as a Markov chain, right? That's why I'm telling you that if you take Professor Kumar's class, he's going to cover, and some of you have taken his class, right? He's going to cover Markov chains in detail. So all of that theory is applicable here, right? So the Boolean model is typical of logical functional models, and many of the issues that arise with it arise in other regulatory network models too. A key issue, of course, is intervention in gene regulatory networks, right? If you find that the network is not behaving in a good way, right? That means it's causing a tumor or a cancer, right? Then you want to intervene and do something about it, right? And this has been mainly considered in the context of a probabilistic generalization of the Boolean model. And that's what I talked about right now, that you will use different Boolean functions with different probabilities, right? And then you try to do the control or the intervention. So the regulatory model which has received the most attention is the so-called Boolean network model. And the model has been studied both in biology and in physics. In the Boolean model, the gene expression is quantized to 2 levels, on and off, right? So it's binary, right? So it's good because we are double-E's, okay? We like computers and binary logic, right? And again, I mean, this is not a new idea, right? How many of you have heard of Norbert Wiener? There are more double E's here. You haven't heard of Norbert Wiener? Yeah, so that's Norbert Wiener. Oh, you were just confused about the first name. See, so Norbert Wiener in the 1940s, right, in 48, he used to be at MIT, yeah, and he's the guy after whom the Wiener filter is named, okay, he's many different contributions. He actually, around that time, he stated that, you know, even in biological systems, right, they would operate with Boolean, with some kind of Boolean logic, or machine-like logic, right? So this is not a new idea that, you know, we just dreamt up in our garage or something, and I'm coming and presenting it here. You know, in fact, he has books on that, and he introduced the area that is known as cybernetics, right? So you can go and take a look at his book from 1948, right, and see what he talks about. Right? But of course, you have to remember in the 1940s and all that, we didn't have all the digital computers and things like that, you know. But he was way ahead of his times. Right? So in the case of the Boolean network, the key defining property is that the expression level or state of each gene is functionally related to the expression states of other genes using logical rules, right? Basically Boolean function rules, right? If, then, else, and so on, right? Although binarization provides very coarse quantization, we note that it is commonplace to describe genetic behavior in binary logical language, such as the gene is on or off, it is upregulated. Remember on the microarray slide we were doing upregulated, downregulated, unchanged. Okay, so all those things are going to fit in now. On and off, upregulated and downregulated, and responsive and non-responsive. So in the context of expression microarrays, which we have already discussed, consideration of differential expression leads to the categories of low expressed and high expressed, thereby leading to binary networks. You know, we looked at the situation where you're looking at the ratio, right? So you are saying it's greater than 1, less than 1, right? Right, or almost equal to 1. So it was ternary, but the methods will be the same, right? So you could have a binary situation where you're saying that if the expression is above a certain threshold, it's upregulated. If it's below, it's downregulated, right? So you could also have the categories of low expressed, high expressed, and invariant, thereby leading to ternary valued networks that are treated in much the same way as binary networks, and often they are referred to as Boolean networks too, right? Now, successful application of the Boolean model requires the inclusion of genes whose behavior is essentially binary, right, or bimodal, right? So it's either 1 or it's 0, right? So the key question is, do you have a lot of genes that are like that, right? And the answer is yes, right? So it has been demonstrated in the context of microarrays that there can be sufficiently many switch-like genes so that binary quantization can be successfully used for clustering and classification. Remember, I said that information on the microarray, right, you're going to use that to design classifiers. So that when a new microarray data is presented, you can correctly classify, right, what type of cancer it is, or correctly do the staging, right? That's what a pathologist does. So you're trying to automate that here, right? And you could even also do gene clustering, right? Now, this is a paper that we had written in 2005. It is available in the literature, right? It appeared in bioinformatics. And in this paper, what we did was, see, like, are you justified in making the assumption that the genes behave in a binary fashion, right? Or that binary logic is going to govern the behavior of the genes, right? So in this paper, we actually worked on data that was produced by a gentleman at the National Cancer Institute, right? Who basically irradiated, right, cancer cell lines, right, using ionizing radiation. So remember, when you go and apply ionizing radiation, you're going to cause DNA damage, right? So the gene expression is going to change. So he had done those experiments with and recorded using microarrays, all right? He had recorded the activity of several genes that are affected by ionizing radiation, right? And he made that data available to us. Now the cell lines that he used, they are in what is called the anti-cancer drug screen of the National Cancer Institute. It's called ACDS, all right? There are 60 cell lines in there. So anytime a new cancer drug comes out, right, you're going to prove its efficacy. First, you have to show on those 60 cell lines, okay, how your drug is working, right? Then you can worry about going to animal trials and, getting FDA approval, clinical trials, and so on, right? But that is like a benchmark, and every year, like I think thousands of compounds are tested. New thing comes out, then this is like the benchmark. Okay, they have cell lines that have been grown from 60 different cancers, right? And that is used like a standard. So he had produced this data, and in this data, right, based on the gene expressions that he gave us, we were able to find many Boolean relationships, right? And I don't even remember what these genes were. Okay, the MRC1 is the OR function of these two guys, these two other genes, right? So similarly, we were able to find lots of different Boolean relationships over there, right? So this Boolean logic for interaction between genes does exist, right? Because otherwise, people can say, are you building castles in thin air, right? So using classical methods, there is ample evidence demonstrating inherent logical genomic decision making, right? And again, I mean, I refer to the work of Norbert Wiener in the 1940s, right? He had said that, and remember, I also talked about when we were looking at the issue of regulation of gene expression, right? I said in the case of bacteria, right? There's one operon that we discussed here. Which was that operon? No, I don't think we discussed the lac operon in detail. We discussed another operon. Anybody remember? So you're not studying for the exam or what? Okay, maybe you'll pull it off at the last minute. Yeah, so we discussed the tryptophan operon. Similarly, there's the lactose operon, right? Because if in the environment you don't have glucose, you have lactose, all right, it can switch on the right genes, right? So it is some kind of a binary logic that is at play, right? So this has been observed even in the 1960s, right? So there are genes that interact with each other via Boolean logic, right? And I'll give you an example. So the figure on the next slide shows a biologically studied regulatory pathway and its corresponding Boolean representation, right? So if you look at this pathway, right? So looking at these names here, can anybody make a guess about what genes we are talking about? Genes or proteins? Just make a guess, right? I mean, we haven't discussed all of them, but do you have some idea about what it is? Or what do you think this RB is? No. RB, it basically inhibits, it serves as a brake on DNA synthesis. So these arrows, all right, that are ending in this kind of stuff, all right, they mean repression, right? That means it's going to stop the following process. And if you have just a link going, that means that is actually an enhancer, okay? So here this is cyclin E, right? Again, do you remember what cyclins are? What are they?
Cyclins are the proteins that bond the microtubule skin, I think. No, that's not right.
No, 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 no. What are cyclins? Anybody remember? Or what are CDKs? All right. Go ahead. Yeah.
Cyclin, doesn't cyclin make AMP?
No. That's different. That's cyclic AMP you're talking about.
Yeah, yeah.
Cyclin is the thing that, whose concentration oscillates. Remember we talked about control of cell division, right? Cyclins and CDKs are cyclin dependent kinases, right? They control cell division. So here if you look at the upper picture, right? Cyclin dependent kinase number 7, cyclin dependent kinase number H. I'm sorry, cyclin H, they interact together, right? Right? Additively, or they enhance each other, and then they will go and interact with cyclin dependent kinase 2, right? CDK2. That is going to interact, see, with cyclin E, right? And then this guy is going to put in an inhibitory effect, right? And then the whole thing then acts on the retinoblastoma gene, right? Retinoblastoma protein, right? So the role of the retinoblastoma protein is to serve as a brake on cell division, right? On DNA synthesis, right? And I discussed that, several lectures ago. So it's there in your notes. As you study for the exam, these things will become clearer. So here, what can happen is that if these factors become active, then they will, release the hold see, the retinoblastoma is acting as a hold on DNA synthesis or a brake on DNA synthesis. Now, if these serve to repress the retinoblastoma, right, protein, then DNA synthesis will start. Okay, and the cell can move into the S phase of the cell cycle, right? So this information you can capture using this logic diagram, right? So here you have an AND gate, right? See, P21 is going through an inverter, so you go through an inverter, and then all these fellows are coming together, and then there is an inverse relationship here because it's a break that you're taking off, so it goes through a NAND gate and you get the retinoblastoma gene, right? So starting from this kind of interaction, you can go to a Boolean network like this. So to summarize, for cells to move into the S phase, right? Again, S phase was the DNA synthesis phase of the cell cycle, right? Cyclin-dependent kinase 2 and cyclin E work together to phosphorylate the retinoblastoma protein, right? At a phosphate group, the shape of the retinoblastoma protein changes, activity changes, right? And inactivate it, thereby releasing cells into the S phase, and that misregulation here can result in unregulated cell growth, right? And potentially leading to cancer. Like the retinoblastoma is a childhood tumor that kids get in the eye, and typically they are dead by the age of 2, right? If somebody has that, has mutations in that gene, then that's bad. Okay, any questions? So, so we have argued that Boolean networks make sense, all right, for studying intergene interactions, all right. So a Boolean network is defined by a set of nodes, V equal to x1 through xn, that's basically the genes, all right, and a list of Boolean functions, f equal to f1, f2 up to fn, right? So each xk represents the state or the expression of a gene gk, where xk is going to be 1 or xk is going to be 0, depending on whether that particular gene is being expressed or not expressed, right? Now, and the Boolean functions, they represent the rules of regulatory interaction between the genes. Okay? Just like I showed you that in that picture, right? Like if you're looking at the retinoblastoma gene, all right, you can write that as a function of those other genes, some Boolean function, right? Then the network dynamics result from a synchronous clock with times t equal to 0, 1, 2, and so on, right? So the value of the gene gk at the next time step t plus 1, right, is determined by this expression. Xk at time t plus 1 is some Boolean function fk, right, for the k gene, which is a function of a number of the activity status of a number of other genes, right? So where the nodes in the argument of fk form the regulatory set for xk, okay, which is the k gene. Is that clear to everybody? Now, typically the number of genes here will be small, right? Because the number of arguments that you have here will be small. Why? Because here you're trying, in this relationship, you're trying to figure out which are the genes or the nodes in the network which directly affect this guy, affect the KH gene. Guess what? In the promoter region of the gene, right, there's limited space. So you cannot have too many factors getting in over there. Okay. Indirectly they might affect, a lot of things might affect it, but directly probably there'll be few, right? So typically here the number of arguments might be like 2 or at most 3. Right. So the number of genes in the regulatory sets define the connectivity of the network, right? With maximum connectivity often limited. So you'll have a big network, but it's the connectivity is not dense, right? It's sparse, right? Each gene is affected only by a few others, directly affected only by a few others, right? So at time point t, the state vector xt is defined to be made up of the activity status of all the individual genes, right? X1 through xn of t, and this has got a name. This state vector is called the gene activity profile, right? Now the functions, the Boolean functions that you've written down for each gene, together with the regulatory sets, they determine the network wiring, right? How the network is wired, right? Which genes, which gene affects, or what are the genes that are affecting a particular gene, right? And you go through all the genes one after the other, right? Now, obviously, a Boolean network is a very coarse model, right? Because you're only allowing binary quantization, right? Nevertheless, it facilitates understanding of the generic properties of global network dynamics, and its simplicity mitigates data requirements for inference. You do not need too much of data to go and infer a Boolean network. Now, if you have a Boolean network, right? So we are looking at the gene expression, gene activity profile, right? Let's say you have n genes, okay? So how many different possibilities are you going to have in the gene activity profile? So there are n genes. Each gene can be binary, right? It can take values 0 or 1, right? So how many possibilities are there for the gene activity profile? Go ahead. I didn't get you. 2 to the power of n. Yeah. So there are 2 to the power of n possibilities. Okay. So there is only a finite number of states. Okay. And if you're cycling through a finite number of states, right? Let's say for all time, right? Then there are states that you will have to visit quite often, right? So after some time you will get into what is called an attractor. You'll keep on repeating yourself, right? Or an attractor cycle, right? So given a starting state, so attractors play a key role in Boolean networks, right? And given a starting state within a finite number of steps, the network will transition into a cycle of states, which is called an attractor. And absent any perturbation, they will continue to cycle thereafter, right? You got into an attractor, you have to stay there, right? Just keep on cycling, right? In something like a limit cycle. Now each attractor is a subset of a basin composed of all those states that will lead to that attractor if chosen as starting states, right? So it has a basin of attraction. If you start from a particular state, you'll keep on going, then you'll end up in the attractor and then you'll stay there forever after, right? Barring any perturbations. So the basins obviously form a partition of the state space for the network, right? Non-attractor states are transient, right? They are visited at most once on any network trajectory, right? So what I'm covering here, this is the deterministic version of what Professor Kumar covers in his course on stochastic systems when he talks about Markov chains and all those different classes and those partial ordering between classes, and then then he's talking about states communicating and things like that, right? This is the deterministic version. This is the easier stuff, right? So he's going to do more heavy duty stuff in his course, putting in all the probabilities and all that stuff. So I'm trying to make the connection here. So for those of you that took his class, you can see what I'm doing, and you can get a feel that the stuff that was covered in that class is going to be useful in this context. For those of you that are going to take his class later, right, you can think of some application area from this course. Any questions so far? So we now have a figure, right? So as you told me that if we have n genes, you're going to have 2 to the power of n states, right? So if n is 6, and that's the example that we are using, so you're going to have 2 to the power of 6 is 64 different states, right? And each state, instead of labeling it by the entire gene activity profile, like this is all zeros, right? I'll just call it by its decimal equivalent, which is 0. Similarly, the other state, all zeros and 1, I'll call it as 1, right? Just for ease of notation, because otherwise every place I'll have to write down all these six numbers, all right? So if you look at this diagram right here, this is the so this is the gene uh, or this is like the transition diagram for the Boolean network. If you start in here, you'll go here, from here you'll go there, then you'll go here, then you'll end up in that attractor 32, right? And that's where you stay forever after, right? You don't come out, right? So here you have three different attractors, 32, 41, and 55, right? And then you have like these four levels. How many steps can you get from here to here, right? So it'll be 4. If you start here, this is level 3, because in 3 steps you'll end up in an attractor. Okay, and the attractors represent the steady state behavior of the genetic regulatory network. So here there are 3 singleton attractors, which I pointed out. There are 4 transient levels, where a state in level k will transition to an attractor in k time points. The attractors of a Boolean network characterize the long run behavior of the network. Agree? Because once you get in there, you're going to stay there, right? Or if you have an attractor cycle, there could be a cyclical attractor, right? Where you don't stay in one state, but there's a number of states that you cycle through, right? But this represents the steady state behavior of the network, so this should relate to the phenotypic behavior of the cell. Okay, because that's what you will see in the steady state. So Kaufman conjectured that this would be indicative of the cell type, because when a cell differentiates, all right, let's say you have a stem cell, all right, that has differentiated into a heart, liver cell, all right, and it doesn't go back the other way, all right, so it's in that attractor and it'll keep cycling, right? So that has been conjectured by Kaufman to be indicated of the cell type and phenotypic behavior of the cell. All right, any questions? So real biological systems are typically assumed to have short attractor cycles, right? Because biology is not going to waste resources, right? So you'll have short attractor cycles, with singleton attractors being of special import. For example, it has been suggested that apoptosis, all right? Remember, apoptosis is programmed cell death, all right? The cell committing suicide. So that's the end of the story. So that should be like, if a cell is in apoptosis, this is not a cyclic procedure, right? If it goes into apoptosis, the cells are dying, right? So it should be going into a singleton attractor, right? Similarly, cell differentiation will also correspond to some singleton attractor, right? You go in there and you're a heart cell or liver cell or skin cell or whatever, right? On the other hand, if you're looking at cell proliferation, right, or cell division, then you have the cell cycle, right? I mean G1, G2, so that could be modeled as a cyclic attractor, And there's a guy by the name of Huang, all right, who many years ago, I mean, yeah, I would say more than 10, 15 years ago, he wrote a paper on this stuff, right? And you can look that up. If you look at our book, right, that we published in 2007 on genomic signal processing, that has all those references, right? So you can look that up. Now, if you have changes, right, in the Boolean functions via mutations or rearrangements, that will lead to a rewiring in which attractors appear that are that could be associated with tumor regenesis. Okay? So in the beginning, you had good attractors, right? Attractors either corresponding to apoptosis, right, or differentiation, right, or you had a cyclical attractors corresponding to cell proliferation, right? But somehow there might be bad attractors that might appear, right? So you have a bad state for the network, right? And if it gets in there, then you have a tumor, right? So in that case, what would you do? You'd like to change the wiring if you can, right? It's difficult. On a cell-by-cell basis, you cannot go in and change the wiring, right? So the idea here is that if you get a cancerous phenotype, right, you might want to shrink the basins, right? Or you might want to do some kind of rewiring. Hopefully that can be done using drugs, right? So that the genetic regulatory network behaves in a nice way and gives you a steady state that is that corresponds to a harmless state, let's say. Okay. So the objective of cancer therapy would be to use drugs to do one or both of the above. Any questions? All right, so now how do you build gene regulatory networks, all right? So this is easy to articulate, it makes sense to us engineers, all right? But how would you come up with all those Boolean functions, or how would you decide, what the truth table is going to be, right, for building which genes interact with which one, right? So for that, one of the possibilities, I mean, there are many different possibilities. One of them is to use what is called the coefficient of determination. And I will try, and this is what is explained in that paper that I'm going to discuss today, right? The use of the coefficient of determination to figure out relationships between different genes, right? So now we've already discussed microarrays, right? So by viewing gene status across different conditions on the microarray, right? It is possible to establish relationships between genes that show variable status across the conditions. If there are genes that go up and down together, right? Maybe there is some relationship between them, right? Or if there is a gene, if this one goes up, that one goes low, there is some kind of inverse relationship, right? So owing to limited replications, we assume that gene expression data are quantized based on some statistical analysis of the raw data. We have already done that. When we did that microarray analysis, right, remember we went through finding out the probability distribution of the density function for the ratio, the confidence interval and so on, right? So that allowed us to do this kind of quantization, right? Now, as you observe the gene expression, right, on the microarray, the variations across different conditions, right, you might be able to find out some kind of correlation between genes, right? If they go up and down together. But again, correlation is looking for linear relationships, right? And it's basically going to be between two genes, right? But you want to do something, a multivariate version of that. And that's what is going to be accomplished using the COD. Now, one thing that I should point out is that correlation analysis, right, does not lead you to causality, right? You don't know which gene is affecting which one, right? You will just know that they are somehow related, right? But you don't know which one is the driver and which gene is being driven, right? So that's an open problem, right? For that, you'd probably have to do some time course experiments, right? And use some prior knowledge and all that, because we have some idea about the signaling networks that are involved, right? So one way to establish multivariate relationships among genes is to quantify how the estimate for the expression status of a particular target gene can be improved by knowledge of the status of some other predictor genes. So if it's gene number 1, right? So gene number 1, gene number 3, and gene number 5, right? So if I think that genes 3 and 5, right, they are strongly related to gene number 1, which is the target in this case, then knowing genes 3 and 5, I should be able to do a better job of predicting what the status of gene 1 is, okay? And this prediction is just like a classification problem, because you can think of genes 1, 3, and 5 having binary statuses, like 1 and 0. So looking at gene 3 and 5, that's like the feature vector, right? I'm going to classify gene number 1 into two groups, right? Either 0 or 1, right? And I will say that they are strongly related, right? If looking at genes 3 and 5 allows me to do a better job, right, of predicting what the expression of gene 1 would be, than I would be able to do in the absence of this side information, right? Is that clear to everybody? Right? That's the main idea, right? So this is formal, and that's what that paper discusses, right? Right? That's what that paper discusses. But you have to find out the best predictor, right? Best predictor without the side information and in the presence of the side information, right? That's how I will compare whether that side information gives me anything extra or not, right? So this is formalized via the coefficient of determination, which is defined in the following way. The COD, or the coefficient of determination, is epsilon sub 0 minus epsilon opt over epsilon 0, right? Where epsilon sub 0 is the error of the best numerical predictor of the target gene in the absence of the observation, right? Don't make any observation on any other genes. Just by itself, maybe based on past history or something like that, you make a prediction, right? That prediction will have some error associated with it, call that epsilon 0, right? Then you basically try to include that side information and predict the activity status of that gene. There is some error associated with that, call it epsilon opt, right? Then the difference of those two divided by epsilon 0, that is called the coefficient of determination, right? So let's look at some extreme cases. Suppose when I include those extra genes in my prediction, that side information, I don't do any better than what I was doing before, then what is the epsilon opt going to be equal to? Suppose there's no new information, right? By putting in those genes, I'm not getting anything extra. Then what is epsilon opt going to be equal to? Epsilon naught, right? Because that was the error I was getting for the best predictor without using any side information, right? So that would be epsilon naught. Epsilon opt will be equal to epsilon naught, which means that the COD is going to be 0, right? Okay? So there's no relationship. Or in other words, there's nothing extra to be gained by looking at that side information. On the other hand, suppose the side information is so good, all right, that when I look at the error with that side information, the error becomes 0. right? Epsilon naught becomes 0. So in that case, what is the value of COD going to be? 1, all right? So this is a number between 0 and 1 with a higher value indicating a tighter relationship, right? So if it's 1, then it's really good. To have the side information, you're doing perfect prediction, right? If it is 0, means you're gaining nothing by using that side information, right? So that's the key idea. Now, the catch here is going to be how would you find out these fellows, all right? The error of the best numerical predictor in the absence of observation, right? So you have to do some kind of optimal prediction, right? Best numerical predictor. Or when you have side information, all right, how are you going to come up with this error, right? That's what that paper is about, all about, right? And I'm going to discuss that next. Right? Any questions on this? So this expression you will see even in that paper, right? And the explanation that I have provided, that is supposed to fill in the mathematical gaps in that paper, because there are steps that are skipped in the derivation of those errors, right? One with side information, the other without side information. So this nonlinear form of this coefficient of determination, what we have defined right now, is essentially a nonlinear generalization of the familiar goodness of fit measure in linear regression. Right? This is nonlinear. Right? This is not a linear correlation analysis. The coefficient of determination of COD measures the degree to which the best estimate for the transcriptional activity of a target gene can be improved using the knowledge of the transcriptional activity of some predictor genes relative to the best estimate in the absence of any knowledge of the transcriptional activity of the predictors. Okay, that's exactly what I've been explaining all along, right? And the COD is a number between 0 and 1, right, which I explained, right? And a higher value is indicating a tighter relationship. And as an example, the following figure shows the COD diagram for the target gene P53. Again, P53 is a gene that we have encountered before, right? P53 basically prevents cells with damaged DNA, right, from getting replicated, right? It's going to halt, it is going to allow for DNA repair, right? It's going to carry out some kind of an arrest in the cell cycle, right? So we're going to look at, and there are lots of possibilities. Let's say you have 10,000 genes, right? And you're going to allow 2 predictors, right? So then you have to look at all the different combinations. 10,000 choose 2, right? I mean, so computationally it will be very, very intensive, right? You might need like a supercomputer or something to figure out which genes will be the best predictors. So the following figure shows the COD diagram for the target gene P53 and predictor genes P21 and MDM2, in which the CODs have been estimated in the context of a study involving stress response. And this study is included in the paper that we are going to discuss, right? So after you go through the method of that paper, right, you analyze that data, right, you compute, let's say you found out epsilon opt, epsilon 0 and so on. Again, see, epsilon opt, epsilon 0, you're not going to be able to find it because nobody is going to come and give you the probability distribution. So you have to get that stuff from the samples, right? The best predictors and all that, if somebody told you what those probabilities are, you can calculate those in closed form. But in the real world, you'll just have access to data. Same as what happened in the classification problem. Right? Remember, we talked about Bayes error and all that stuff. We cannot, get a handle on the Bayes error. So then we have to estimate it from data, right? If you estimate it from data, you need large sample sizes, right? So then we went to constraint classification, right? For smaller sample sizes, we said that was better. Same thing will happen here. This is prediction. It's just like classification. Exactly the same thing is going to be. Same considerations are going to play an important role. So here you have P53, right? If you try to predict P53 just using P21, the COD that comes out after the calculation is 0.227, right? If you just try to calculate P53 using just MDM2, this gene MDM2, the COD you get from analyzing the data is 0.259. However, if you use both MDM2 and P21 together as joint predictors, then the COD goes up significantly. It's 0.452. Okay. So in this figure, and again the calculations, you will understand how to do that by looking at the paper. And if you have the time, you can go run through that. Even software is also available for doing those kind of calculations. So we see that the individual CODs for P21 and MDM2 are 0.227 and 0.259 respectively. But when you use them jointly, the COD for the predictor set it jumps up to 0.452, right? Now biology, so again, I mean, this was work that was done like in 2000 or so, right? And it's a question of applying signal processing methods to find out relationships between genes. Okay, and the hope is that from the data you will pick up relationships that biologists know, and you'll pick up more, right? And that's what actually did happen. Otherwise, we would not be teaching that here, right? And that's what started a lot of the collaborations between A&M uh, and the biologists that we have collaborations with. Right? Yeah, go ahead.
So essentially, you can use this to map the network of the activations of the genes.
Yeah.
I mean, is there any way to do that in like a repeatable algorithmic format? I mean, you say that you have like 100 genes or something that you might question cause a certain disease or something, and then you look at the mappings, and I guess you could find the best mapping with this method to that sort of thing.
Yeah, but it is still an exhaustive search.
Yeah, it
Yeah,
also,
yeah, you could, yeah. You could. That's what they tried to do.
that's what they do. Okay.
Yeah, that's what they tried to do. Okay. See, but again, I mean, here, remember, I think you can do better than this because there is prior information, right, about which gene affects which one. You can stick in some of that information. That will hopefully cut down on the computational effort. Yeah, you can do. And again, in biology, one thing I have to mention to you is that even if you come up with a method, anytime new data is generated, that's a new paper, right? Or if you generated data and you published it in some paper, right? With the same data, you cannot go and publish again, right? It's not like in engineering where, if you write that grand old paper that covers everything, then you're out of business, right? Here, if you do gene expression data from mice, it's one paper. If you do from human cell lines, it's one paper. If you do on canines, it's one paper, right? So, but it's very, very labor intensive because there are a lot of experiments and people have to do the work. You cannot just sit down with a piece of paper and pencil and just write some theorems and get papers out. So the culture is different, right? So biologically, it is known that p53 is influential but not determinative of the upregulation of both p21 and MDM2. Right? That means the COD is not 1. Right? So hence, it is not surprising that some level of prediction of p53 should be possible by a combination of these two genes. Right? These genes are driven by p53. Right? p21 and MDM2, they are driven by p53. In fact, there is a loop. Right? And we have another paper that looked at relationships between these genes. Right? And how their expression patterns oscillate, right? So note that the prediction of p53 by p21 and MDM2 results from the regulation of p53 on them because p53 affects both those genes. So going the other way, the same study also found the COD for p53 predicting p21 to be 0.473. This is not a linear relationship. If it's correlation, both ways it will be the same number, right? Here this is not, it's a different number from what we got the other way around, right? Now the increased predictability of p53 using both MDM2 and p21 is expected because increasing the size of the predictor set cannot result in a decrease in the COD because you're using more size, unless that information is completely garbage, right? It should not decrease the COD. So in the figure that we showed a few slides back, MDM2 and P21 have got very similar CODs relative to P53, and there is a significant increase when they are used in combination, right? I highlighted that, right, when we were looking at the figure. On the other hand, it may be that very little, if any, predictability is gained by using predictors in combination, right? Because the side information you are bringing in might not be any new information, right? So you'll gain nothing. So moreover, it is possible that individual predictors have got CODs that are very close or equal to 0. Right? By themselves, they are very poor, but when used in combination, the joint COD is 1. This is similar to the AND gate. If you look at one input at a time, it's very poor predictor of the output. But if you take them all together, right, you can predict the output well. So this kind of situation shows that it is risky to assume that a predictor G1 and a target G0 are unrelated because the COD of G1 predicting G0 is very low, right? Because in combination with something, it might produce a strong association. So this situation is akin to that in classification, where a feature may be poor if used alone, but may be good if used in combination with other features. And the issue in both settings is the danger of marginal analysis, drawing conclusions about variables from marginal relations instead of joint multivariate relations, right? So yeah, the next topic we'll move on to is probabilistic Boolean networks, all right? But before we move into that, I think this is the appropriate time to discuss that paper on COD, all right? And another thing that I want to point out is that this course at least gives you pointers towards important classes that you should take within electrical engineering. One of them was Professor Kumar's class on stochastic systems. He's a guru in that area. He's written books in that area, I think in 1986. All right, so it's best to learn from the guru. Okay, so go and take his class. I sat in on his class last spring. Right, I had to miss about a week of that because I came down with the flu, but it was really worth it. Okay, because I mean he's a very good teacher too. So consider taking his class. And again, I mean that will probably fill up. So be on the lookout. Another class where you learn about all this feature selection and all these things that I'm talking about is 649, right? Pattern recognition. So these are classes where you can build up the engineering expertise so that you're better equipped to work on biological problems, whether they are in medicine or in agricultural applications. Remember, without those skills, you're nowhere, okay? Because I mean, if you don't know the double E stuff, right, you cannot go into a new field and you're going to apply your tools. Nobody's going to come and tell you, hey, this is your problem, okay, use this tool. You have to be able to figure out what to use, right? Okay. So, I mean, some of you come to me for advice about course selection. I'm giving that to you right now, well in advance. And these are good suggestions. But again, I mean, nothing is mandated, but I'm just pointing out from the kind of problems that we are seeing in this course. Okay, so now we are going to switch gears, all right? And, before trying to finish the rest of this chapter, right, we are going to switch gears, go to that paper on COD, all right? Go through it step by step, just like I went through the ratio analysis paper, all right? And try to get a good understanding of what is involved. Right? So any questions so far before I make the switch? All right, so let's start talking about this paper. All right, that's paper number 2. All right, that is posted on eCampus. So this is a general nonlinear framework for the analysis of gene interaction via multivariate expression arrays. All right. This is the engineering version of the paper. All right. The same paper with more biological explanation and all that is available, and I believe it's referenced here. Right. So let's take a look at the abstract. All right. And you'll see, and you'll be pleasantly surprised to see that a lot of the stuff you already know. So it says a cDNA microarray is a complex biochemical optical system whose purpose is the simultaneous measurement of gene expression for thousands of genes. Okay, nothing new here. You guys already know that. We discussed the ratio analysis paper. We discussed microarrays. So in this paper, we propose a general statistical approach to finding associations between the expression patterns of genes. That's exactly what I told you that this paper does. Right? So we find association between the expression patterns of genes via the coefficient of determination, right? The COD that I already introduced, right? So this coefficient measures the degree to which the transcriptional levels of an observed gene set can be used to improve the prediction of the transcriptional state of the target gene relative to the best possible prediction in the absence of observations, right? In the absence of what I refer to as side information, right? The method allows incorporation of knowledge of other conditions relevant to the prediction, such as the application of particular stimuli or the presence of inactivating gene mutations as predictive elements affecting the expression level of a given gene. Like, instead of making the activity status of a particular gene depend only on the activity status of other genes in the network, right? You could say that it could depend on some other extra factor like the temperature or something like that. You can put them in as additional variables, right? So various aspects of the method are discussed: prediction, quantification, unconstrained prediction. I talked about this, right? Then constrained prediction using what are called ternary perceptrons. We'll discuss that. And design of predictors given small numbers of replicated microarrays. The method is applied to a set of genes undergoing genotoxic stress, right? You subject the cell to some stress, like I mentioned about ionizing radiation. You could subject it to different stresses, and the activity patterns of the genes will change. Right? That's going to give you more data to work with. Right? So this method is applied to a set of genes undergoing genotoxic stress for validation according to the manner in which it points towards previously known and unknown relationships between the genes. The entire procedure is supported by software that can be applied to large gene sets, has a number of facilities to simplify data analysis, and provides graphs for visualizing experimental data, multiple genes, gene interaction, and prediction logic. Right? So this is very much the description that I gave you, right, as we built up towards this paper. Right? So now, let's start with the introduction. Right? You should have absolutely no trouble with this introduction because all these terms, right, the microarrays, then gene expression, right, and things like that, you are already familiar with this, right? So there should be no trouble here, right? So you're trying to find out relationships between genes. The mechanism of intergene association is not a factor in statistical prediction because this is like a correlation analysis. You are not looking at causation, okay? What is causing what, right? So this is a multivariate generalization of correlation analysis, right? So that's what he's mentioning over there. So as the first step in carrying out nonlinear genomic prediction, right, you're going to do the quantization, right? So either genes are high or low, right, not work with analog signals. And we know if we use the ratio analysis paper, we have a ternary quantization, right? 1, 0, minus 1, right, for each gene and work with that. So now looking at the activity of each gene, right, either 1, 0, minus 1, right, can I use the activity of some other genes to make a prediction for this one, right? And how good is that prediction? That's the key question that you're trying to answer, right? So here we are going to do nonlinear multivariate prediction, right? So the problem is the following. If you look at this picture, right? So these are the inputs, all right? So these are the inputs that are going into the predictor, right? Based on these inputs, you want to find a function that is going to spit out. So let's say x1 through xm, these are some genes, right? That you activity or expression patterns of some genes. You want to use this information. Looking at this information, you want to make a prediction for some other gene, right? Y predicted, right? And the question is, how does this, and how good is your prediction? That will depend on what the actual value of Y is, right? So you want this prediction to be close to this Y in some sense, right? That's what you're trying to do. And there might be additional variables that are going in that are latent variables that you cannot measure. You don't have any control over, right? So the question is your predictor going to be good, right? So, and again, I mean, this function here is not known, right? What this function is going to be, that's not known, right? So you want to come up with this function, right? Now, you can do, see, if I said that we are going to quantize the expressions, all right, to ternary levels, all right, 0, 1, and minus 1, all right. So if you're going to do that, okay, then x1 will have three possible values, 1, 0, minus 1, x2, three possible values, 1, 0, minus 1, and y predicted also will have three possible values, 1, 0, minus 1, right. So the most general case would be a truth table, right, and I'm looking at the case right now, just as an example, let's say we have only two predictors, x1, x2, right. So if you're going to construct a function, then you need a truth table that is shown here, right? So you have x1, right? All the possible values for x1, right? 1, 0, minus 1. All the possible values for x2, 1, 0, minus 1, right? So how many total number of possibilities do you have for x1 and x2 together? 9. So there are two predictors, all right? M is 2, and you know, for each predictor there are 3 possibilities, so 3 squared. That is 9 possibilities here, right? Then, you know, the truth table in each location, right, y1, right, there are 3 possibilities, right? I mean 1, 0, minus 1, right? In the second location also 3 possibilities, 1, 0, minus 1. So if you're looking at this truth table, right, how many possible truth tables can you have? How many possible, like how many possible different, you know, truth tables, you know, capturing different functions. See, here we are not putting in an explicit function, right? It's the truth table. If x1 is minus 1, x2 is minus 1, then this y predicted, all right, the value that you are getting here, all right, it can have 3 possibilities, right? 1, 0, minus 1, right? It's not 27, no, no. It's 3 raised to the power of, yeah. So 3 raised to the power of 9, right? So that's a pretty large number. Large number of truth tables, okay? And you, so to estimate from there, I mean, from this large number would be difficult, all right? On the other hand, if somebody says, all right, y is, let's say, a linear function, right, of these two variables, all right, x1 and x2, right? So then y is like ax1 plus bx2, right? Then all I need to find out this relationship y is just those two parameters, A and B, right? It will require a lot less data, right? As opposed to trying to estimate the different entries for all those 3 to the power of 9 possible truth tables, right? But the linear one might be too simple. So we'll use something that's a little bit more general than the linear one, right? So we'll have a linear thing and then that is going to be thresholded. Okay, that is called a perceptron, right? Where like, so you compute like some linear combination of let's say x1 and x2, ax1 plus bx2, you get a number, right? Compare that with let's say with 0, right? If that is above 0, then you say something is 1. If it's below 0, you say it's 0. Or maybe you set some interval, some minus zero point 0.5 to 0.5, right? So if it is greater than 0.5, I call it class 1. If that linear combination lies between minus 0.5 and plus 0.5, I call this 0. If it lies below minus 0.5, I call this minus 1, right? I still have to estimate some of those parameters, that A, B, and all that, but the number of parameters to be estimated will become a lot smaller, right? So this is an example of constraint prediction, right? If you go with the truth table, that is unconstrained prediction. The only constraint is you did the quantization, right? But beyond that, I have not put any constraints, and you can see it is going to require a lot more data, right? But because I will typically be operating in the range where that much of data is not available, right? I'm going to do constraint prediction, and the constraint will be imposed by working with what is called a perceptron, right? See, like here, y predicted is some nonlinear function g. It's a thresholding function of a linear combination of these fellows, right? Right, and this has been used. These perceptrons, they predate what are called the neural networks, right? Some of you may have heard of that. In neural networks, what is going to happen is you're going to take the variables, take a linear combination, pass it through a nonlinear operator, right? You get some outputs, all right? Again, do the same thing, all right? You basically take different linear combinations, again pass through nonlinear operators. So it is like structured nonlinearity, okay? And the reason why people like to use neural networks is because somebody has shown that if you have a neural network with enough layers and enough number of nodes, you can approximate any continuous function, right, to an arbitrary degree of accuracy on a compact set, right? So it's a good fun, but there is no theory that tells you how many nodes you need to use, how many neural networks and all that stuff, right? So and then you try to learn all these different weights and these different weightings in the linear combinations. So this is a simpler version of that. You don't want to use neural networks because neural network will again be having too many parameters. See, being able to fit any arbitrary function does not come for free, right? There will be a lot more parameters, and you don't have enough data to learn all those parameters. So you will work with the perceptron. Okay, so in this paper, that's what they are working with. Uh, Now, before that, there is the same discussion, okay, about the error of the optimal predictor, epsilon op. That's like your Bayes error, all right? But the problem is in the real world, you don't have the probability, so you will end up with actually what you can get from the samples, from the sample that you have at hand, all right? And then there is a problem if you don't have enough samples, all right? The error is not going to be small enough. So what do? You do constraint prediction, right? And the constraint is imposed using a perceptron, right? So that's stuff that you've already been exposed to before. This is that same picture that I showed you in the context of classification, right? So this is like the Bayes error, right? If you were doing classification, but this is the error of the optimal predictor without any constraints, right? If you were using that complete truth table, right? Now the problem is that the expected error that you will get, that goes down with the sample size, right? But you will typically be operating in this region because you have small number of samples. So I will not use the unconstrained predictor. Instead, I will use the constraint predictor, right? Use like a perceptron. That's a constraint predictor. Problem: this error, the best that I can do using the constraint predictor is usually worse than I would do. The error is going to be higher than that of the best unconstrained predictor, right? Unless the best unconstrained predictor also happens to lie in that constraint class, okay, which is usually rare. So then when I do the estimation, right, when I try to estimate the best constraint predictor, then the error that I make is lower than the error that I would get using the best unconstrained predictor. So I typically will operate in this region, right? So these are all things that I have already discussed, right? So the bottom line is if you're now going to calculate the COD, right, I need to be able to get a handle on epsilon, right? Epsilon n, right? Epsilon constraint optimal n, right? And then use that formula. right? Epsilon dot minus epsilon opt divided by epsilon dot. That's what I had in the notes. Okay, I'm going to use that formula, right? So any questions so far? I'm trying to communicate as much information as possible about this paper to you, but if you don't take a little bit of time and read it, right, I cannot teach you all of this stuff. I'm trying to show you the ropes, but you have to spend a little bit of time. And I'm going to show you more. Wherever I thought you'd have difficulty, I'm going to work that out in that additional handout that I gave you, right? So this discussion is all the same as what we had before, right? This thing, in the context of classification, it's already discussed in those PowerPoint slides, right? So now you got the perceptron, right? And he talks a little bit about the neural networks having multiple layers, so let's see G1, G2, and so on, right? But we're not going to use it because you will need a lot more data to learn neural networks, right? For other applications, you probably can, right? So now we come to the coefficient of determination, right? So that is going to be obtained, right? Once I've figured out what the estimates for those errors will be, right? With and without side information, then I will plug that into that formula, right? And I will calculate, right? So before I can calculate these errors, I need to be able to find out the best predictor without side information and the best predictor with side information. Then I can plug in the numbers here. First, based on the theory, I will compute that. Then I will calculate this based on sample data. So this will become functions of n. See? Do you see that here? Right? That's what they're doing in the paper because you will not have the distributional knowledge and all those probabilities to compute this in closed form. Right? But I will give. But he does mention about computing it in some special cases where it can be computed in closed form, and that's what I'm going to present to you here. Right? Let's see. Yeah, you can see that in the closed form, he's getting some stuff here. All right? The COD, you can, in some special cases, if you make some assumptions about Gaussian and things like that, right, you can calculate. And that's what I want to highlight in that explanatory material, right? Okay. Like the COD, see, he shows that the COD is something like the square of the correlation coefficient, right? So in that special case, in the case where you have Gaussian distributions, right, or the random variables are jointly Gaussian distributed. Okay, any questions? So now I'm going to go to the explanatory material, right? So we've come to the stage to equation 5 where we need some explanations, right? Some gaps have to be filled in, and even the derivation of this one, right? So let's go to that explanatory material. That's the other handout that I gave you. And again, this is material that you will pick up in EE courses, right? There are probably courses on estimation theory, stochastic stuff. They should be covering these, right? I'm covering them here because, I don't know how many people have had those classes, and I don't want you to get lost because you don't know that stuff, right? So this is the explanatory material. Now, first fact, right? Given a real valued random variable y, the best constant predictor of y in the absence of any observation, right? No side information, right? Is the mean of y, right? So why is that? Because you look at the mean square error, right? And you will see that so for a constant predictor c, the mean square error is given by y minus c square, right? So you squared the error, then you took the expectation because it's a random variable. So then you square inside the expectation, you get y square minus 2cy plus c square. Write this out. Now we are going to try to minimize this, all right, with respect to your choice of the constant c. So differentiate set derivative equal to 0, right? You get c equal to the mean of y, right? You take the second derivative is positive, so this is a minimum, right? So thus c equal to the minimum of y minimizes the mean square error, right? If you're using a constant predictor, right? Agree? So now if you do have data, okay, if you have side information, then it'll be the mean, but it'll be the conditional mean, right? And that's, I'm going to show you the derivation over here too, right? So the minimum mean square error estimate of a random variable y given an observed random variable x is the conditional mean of y given x. That is the expectation of y conditioned on x, right? And again, this does not make any assumptions, right? So if you have the probability, if you have the conditional density function, you can calculate the conditional mean, and that is the best estimate, right? But it requires knowledge of the conditional probability density function, right? Which might be something difficult to get, right? Yeah. except in some few cases, like if the random variables are jointly Gaussian, then you can get that. So let's do the proof of this. Right. So we want to now prove that the best estimate in the presence of observation is the conditional mean. Right. So you start again. You're looking at the error of the prediction of y, which is y hat minus y square, right? The mean square error, right? So again, do the same thing as before, right? So you can write this as. So now we do a little bit of cheating, right? Because we know the answer. Okay, somebody has told you that it is a conditional mean, right? So I'm going to just add and subtract the conditional mean of y conditioned on x, all right? So y hat minus expected value of y conditioned on x plus expected value of y conditioned on x minus y square, right? Now I basically write this out completely. So the first two terms here, I basically square that, right? Then I will have the last two terms, all right? Again, that's squared somewhere, yeah, here, right? Right? And then you'll have the cross term, which is going to be 2 times the expected value of y hat minus expected value of y conditioned on x multiplied by this other guy. Okay? So now we are going to focus attention on this fellow, right? I'm going to show you that this fellow is equal to 0, right? And how do you do that? Well, when you calculate the expectation, right, you can condition so you have a number of variables, right? You can treat some of the variables as non-random, right? Calculate the expectation and then average out the randomness over the variable over which the expectation you didn't take earlier, or over which you carried out the conditioning. This is a trick that Professor Kumar will also use several times in his class, right? If you remember when he was trying to show the optimality of some of those policies, all right, this conditional expectation was brought in, okay, because you don't change anything, right? So here you have this term, the expectation of this quantity. Now instead of taking the expectation over all the random variables, I'm going to condition the expectation of x on x, okay? This is still a random variable because I've been treating x like something that is not random, but really x is random. So now after that I need to also take the expectation over x, okay? So I condition it on x and now I'm taking the expectation over x, so I will get back exactly what I had before, right? So now when you're conditioning the expectation over x, okay, look what happens. See, y hat is a function of, y hat is the estimate of y. It's a function of what? Huh? Huh? It is, yeah, so the data is x, right? I mean, I'm looking at x, all right? Yeah. So data is x. So if you're conditioning the expectation on x, all right, I don't need to take the expectation of y hat. Y hat will just come out of the expectation, or the inner expectation. So y hat comes out. Even the expectation of y conditioned on x, all right, that's a function of x because you averaged it out, or you took the expectation treating x like a non-random variable. So that's a function of x, all right? And so since this is conditioned on x, I can pull this guy also out. So this factor can be pulled out of that inner expectation, right? And what you're left with is expectation of the expectation of y conditioned on x minus y, the expectation of that whole thing conditioned on x. Now if I evaluate this expectation here, all right, see you have expectation of y conditioned on x, all right? You're again taking an expectation conditioned on x. So what is this term going to give you? So you have expectation of y conditioned on x, but you're taking again an expectation that is conditioned on x. So you will get back exactly the same thing, right? Because here all the other randomness has been averaged out, right? But then even this additional expectation also is conditioned on x. Okay, x is not being treated as a random variable, right? So you'll just get expected value of y conditioned on x, and then the next term that you get is, because expectation is a linear operation, is minus expected value of y conditioned on x, right? So these two terms will cancel out, and so here what you get is 0. So this term drops out, right? This term drops out, and therefore the mean square error is basically this first term. It's a sum of two squares, right? It's the sum of this square plus this square, right? Now you're going to choose the y hat, right? That minimizes the sum of these two squares. Look, the second one, right, does not depend on y hat. It doesn't matter how you choose y hat. That's a constant, right? So the only freedom you have is in choosing the first one. So if you choose y hat equal to the conditional expectation, right? Then you will have the minimum value, and the minimum mean square error is what you get here. Okay, agree? Okay, so this is something that they will cover in your estimation class, right? I think let's stop here for today, right? So this is without, so this one doesn't make any assumptions about Gaussian or anything like that, all right? Among the class of all possible predictors, the best one is the conditional expectation. Next time I will show you that if you make some assumptions, then you can do better, you'll get a linear predictor and so on. Okay, so let's stop here for today.